the university and to the whole organisation for inviting me here. It's really a very great pleasure. My first time in Nijmegen, and I hope I can live up to that introduction. It was very nice of you. And I don't usually start with apologies, but I will make two little apologies. The first is that even though my name is Hollander, I won't be speaking in Dutch. I apologise for that. Maybe one day I'll learn some Dutch. And the second is I've got a lot of slides, a lot of ideas I want to convey to you, and so I'll be going quite quickly in English through all of these ideas, and I hope I will make them clear to you, but if there's anything that's not clear, then there'll, I think, be plenty of time for questions at the end and for clarification. So, although the title is The Future of Regenerative Medicine, and I will be covering that as an issue as we go through, most of what I'm going to talk about inevitably is where we are at the moment, what we can do at the moment. Let me start with a couple of definitions to make sure we all understand what we're talking about. Regenerative medicine as a concept is about trying to replace those parts of the body that have become diseased, that have become dysfunctional, damaged for one reason or another, and to try and do that not in the traditional way with drugs, and drugs tend to slow down processes but don't tend to regenerate organs. So instead of using drugs, we think about doing that using cells, the cells of the body as the powerhouse for driving repair. And we can think about stimulating cells inside the body, but more usually for regenerative medicine, we think about implanting cells, either on their own, for example, stem cells, which I'll come to in a moment, or cells combined with some kind of scaffold, a three-dimensional biomaterial that's compatible with the body and that can give shape and deliver the cells in a way that's appropriate for the particular organ being treated. And some of us call that tissue engineering, this combination of cells and scaffold to give shape. What do I mean by a stem cell? Because I'll talk about stem cells quite a lot during the next 45 minutes. Most of the cells of our bodies, as they grow and divide, they age. Just as we age, our cells age. And so when they've undergone a certain number of divisions, they will die, they'll die out. But stem cells have a tendency to survive much longer. It depends on the kind of stem cell. True stem cells, if you like, will continue to divide without ever aging, and that means we can use this property, which we call self-renewal, very effectively in the laboratory to grow up a large number of cells, and that makes them very useful to us. Some stem cells only grow a certain amount and then they die, but still they will grow more than most of our body's cells. The other property is called differentiation, which is when a stem cell which has no particular function turns into another kind of cell which does have function. So it might become a liver cell, or a heart myocardial cell, or a cartilage or bone cell, and do the work of those tissues and organs. And so this combined property of self-renewal and the ability to turn into lots of different cell types makes stem cells very interesting to us in regenerative medicine. Size matters. Why have I put that there? Well, we're talking about the future of regenerative medicine, and one of the issues we need to think about as we look forward in regenerative medicine is how big an organ, how big a part of the body we can create in the laboratory. How much complexity can we build into those organs as we grow them. So there are some tissues which we can grow relatively easily. Nothing's easy in this field, but relatively easily already. For example, parts of the body that are flat sheets. I'll come to examples in a minute. Relatively thin and simple. Or tubes, like the trachea, like the airway, that I'll come to in a while. So those are relatively easy, and the reason they're easy because these tubes or those sheets are thin, and oxygen diffusion and nutrient diffusion through them when they're growing in the laboratory or soon after implantation is relatively straightforward. We can also use regenerative medicine to heal tears in simple tissues, and I'll give you an example of that in a moment, to bring tissues back together. What we're really not very good at doing at the moment is creating solid organs, liver, kidney, heart, for example, and there are a number of reasons around that, which I'll come to towards the end of my talk. And I think a lot of what we need to think about is, can we use all the learning and the skills we develop for these relatively easier problems in order to find ways of creating big solid organs with a lot more complexity. So I'd like us to think about that this evening. So for the next 40 minutes, I'll give an example of sheets of tissue, these relatively thin tissues. I'll give an example of closing gaps, how we try to remove space, as it were, to repair using stem cells. I'll give one or two examples of tubes that can be used, that can be grown in a lab and implanted, and then at the end I'll look forward and see if there's any reality to anything more complex. A lot of what I talk about is cartilage. My main area is cartilage. It's not all I'll talk about today, but it's the predominant tissue that I have experience of. I've spent all my scientific life trying to understand this tissue, which appears to be very simple because it only has one type of cell in it. We call it the chondrocyte. It's a cartilage cell. It doesn't have a blood supply and it doesn't have a nerve supply, so it's just one cell type. And that cell type makes all of these different proteins, like the collagen shown here, and this protein glycan, which binds water and makes cartilage act a bit like a sponge to protect the ends of the bones. And so if we're going to engineer cartilage in the laboratory, we need to be able to reproduce this kind of structure, and we need to have ways of measuring that structure and so that we can be sure we've reproduced it before we implant that tissue into the body. So a lot of what we do in my laboratory is figure out really good ways of measuring the quality of this tissue as we produce it, as we create it, so that we can be sure that we're going to implant something that's functional and as similar to the natural tissue as possible. Okay, there's one or two videos in this presentation, and those of you who aren't used to seeing surgery might want to look down between your knees and close your eyes. The next slide is the most gory, all the others are relatively okay, I think. What I'm going to show you now is an example of an early cartilage tissue engineering technique which was developed in Italy by a company called Fidia, where the patient's own cartilage cells were used to treat cartilage damage in the knee. And those cells were grown up in a clean laboratory and then seeded onto a membrane, a three-dimensional scaffold. This one was made out of something called hyaluronic acid. For those of you who are material scientists, esterified hyaluronic acid. And what you're going to see is one of the very first patients in whom this is being implanted. So close your eyes now if you don't want to see blood and gore. So here is the knee being opened up, and here is the cartilage lesion there. And here the surgeon is cleaning up the cartilage lesion down to the bone, which is the slightly bloody surface down there. And then you'll see that the metal packet that the tissue engineered product has been carried into the laboratory is used as a template. It's cut to the shape of the lesion. And then that template is put onto the tissue engineered product, which is cells on scaffold. It's not a very mature cartilage. It's been grown for about 2 weeks. And that's cut to the right shape. And this material is quite sticky, so it doesn't need suturing, which is very useful. So it's kind of shoved in. And then, are there any orthopedic surgeons here? I always like being rude about orthopedic surgeons. So then the orthopedic surgeons, who are very sophisticated people, they do this very clever trick to see if it stays in place. And that's open surgery, and because there's no suturing involved, here you can see another lesion. This technique can be performed down an arthroscope, that is a tube looking into the knee, so much better for the patient, much quicker recovery, and this is essentially the same operation, but without fully opening up the knee. And what you'll see is for this particular patient, these are put in place, and then the next video, there it is, here is the same patient 1 year later, and you can see that all that space has been filled with something which, when the surgeon compresses it, has mechanical resistance and looks like cartilage, and biochemically we've analysed that, and histologically, and it has a fairly close resemblance to natural cartilage. Now that tissue, you couldn't really see it there, that's about 2 to 3 millimeters thick. So when I talk about thin sheets, that's about as thick as you get. And there's a number of different techniques for repairing cartilage, all with similar degrees of success, which is only really partial success, but nevertheless you can get the tissue to grow relatively easily, you can get it to stay there, and even 10 years later, many patients treated with this kind of technique will have a pretty good cartilage repair. So that's an example of a sheet, and just showing you that this kind of technique or similar approaches are used already in the clinic around Europe and around the world. We don't want to stop there, because that can only be used for a limited range of patients with traumatic injury, like athletes who injured themselves. We would like to be able to treat early osteoarthritis. This is a man whose joints have worn away, far too late to be treated with regenerative medicine, I would say, and he'll do very well with an artificial knee. But there are people in their 40s and 50s who are on their way to this kind of disease, and we believe that if we can get in early with a cell-based therapy, with regenerative medicine, then we may be able to prevent this, or at least delay it by 5 or 10 years, and we think that that would have a huge benefit for the quality of life of the patient, and probably for health economics as well, the cost of looking after these people. But the challenge is huge, because the lesions that you end up with are extensive and there's really not much cartilage left, and so we think that for this we're going to have to engineer cartilage that's already mechanically competent, that is, it already works before you implant it. The video I showed you, the videos, were cartilage that wasn't really properly formed, it was just cells on a scaffold implanted, and then the growing happens over many months in the body. But to treat even early versions of these kind of lesions, we think that you're going to have to create fairly large pieces of cartilage that's already quite mature, and to do that we think we need stem cells, and we have a pretty good source of stem cells, which is the bone marrow, which is rich in a particular kind of stem cell called the mesenchymal stem cell. Don't worry too much about the name. When I say rich, it means we can get them out of there. There's not that many cells in the bone marrow, but when you coat them onto plastic, they stick down and you can grow them up, and there are many groups around the world which do this kind of thing. And the idea is to take bone marrow from what's called the iliac crest, from the hip there, isolate these stem cells, seed them onto a scaffold, and then grow cartilage. And we spent a number of years showing that we can do this. We can produce pieces of cartilage even from quite old patients with advanced osteoarthritis. And I'm not going to take you through the histology or the biochemistry, but we think that we can make relatively good cartilage with this kind of technique, and we're now working on methods to try and enlarge these so that we can create even bigger sheets, and I'll come back to that at the end of my talk. So that's sheets. I talked about closing gaps as well, if you remember, and that gives me a chance to talk about the work of my spin-out company that you mentioned, which is a company called Azalon. So as a conflict of interest statement, let me say that this is a spin-out company of which I'm a co-founder and a chief scientific officer. I'm showing you this data because I think it will be of interest to you in the context of this talk. So the company formed around a discovery we made when we were trying to figure out, when we've engineered cartilage, how we implant it and get it to integrate with the surrounding natural tissue. How do you get one piece of cartilage to integrate with another piece of cartilage so that they stay together and attached for a long period of time? And this was quite a long project. It took us quite a while to figure out how to do this. And we ended up with this thing we call a cell bandage, which is essentially a membrane. In this case, it's at the beginning we were using something called polyglycolic acid that some of you will know about. Don't worry too much what that is. It's a synthetic membrane. Now we tend to use collagen scaffolds. And we end-seed that membrane with cells, and we started doing this with cartilage cells, but now we use stem cells, which we haven't turned into any other cells. So undifferentiated mesenchymal stem cells. And the idea is that these cells migrate between the two surfaces, and in this model, this is cartilage where we pulled out one piece and pushed it back in with the cell bandage in between, and the cells, we think, will migrate between the two surfaces and drive a repair process over a relatively short period of time. And this is one of our first experiments. On the left there, you can see a control where we just had the membrane without any cells, and there was no integration at all. In fact, there was probably degradation. And here, where we had cells included with the membrane and the cell bandage, you can see there seemed to be some kind of healing across that interface. And histologically, if we pre-label the cells so that they're red, you can see them migrating from this membrane into the cartilage. There's some cells there in the process of crossing over, and you can see them here as well. And this border between the membrane and the cartilage gradually metabolizes and disappears, so we get this effective integration, which we can test by mechanically trying to pull it apart. So that technique, cell bandage, was developed to try to figure out how to implant tissue-engineered cartilage into the joint, and we're running studies in a sheep model now using that technique. And early signs are it's working, but I don't yet have data to show you. But the early signs look quite good. But we realized that this approach of closing gaps between tissues, joining one tissue to another, could have many applications in medicine. Is it really regenerative medicine? Well, it kind of is, because I think we can use it to restore tissues and organs to their original state. And in particular, we became very interested in using this for a particular kind of cartilage called the meniscus. In your knee, and only in your knee, there are two pieces of fibrous cartilage, which are called the meniscus, that sit in between the bones. And if you hear of an athlete or a friend who have torn a cartilage, it's this that's gone, and you can see an example of a tear there. Look, this is looking from on top, looking from in front. You can see you have one on one side, one on the other. If you tear a meniscus, it causes a lot of pain, and surgeons used to remove the whole meniscus to take the pain away, and that worked for a few years, and then the patients all developed osteoarthritis. So if you were 20, 21 when you tore your meniscus, by the time you were 30 you might have advanced osteoarthritis. Not a good outcome. Relatively more recently, the surgeons have become a bit more refined and only remove the damaged part of the meniscus, but even then there's a high risk of osteoarthritis, at least 50% within 5 to 10 years. Our idea was to take this cell bandage, these stem cells, which are undifferentiated, they're still stem cells, on a collagen membrane, and put them into the middle of that tear, and then sew the tear up around the cell bandage. And the hypothesis was that the cells would migrate between those surfaces and heal that lesion. Then there would be no need to remove the meniscus, and we hope and hypothesize that that would avoid the development of osteoarthritis. So this was one of our first in vitro experiments, where we used human stem cells, mesenchymal cells, on the collagen membrane implanted between two pieces of sheep meniscus. And we undertook some sheep studies. Professor Alan Goodship at University College London did those for us, creating a meniscal tear with implanting the cell bandage into this model. And we were able then to distinguish those animals where the tear healed up. You can just see the suture there in blue, and those controls where the tear was still in place 3 months later. We formed a spin-out company, as I told you, Avalon Cell Therapeutics, excuse me, and we did a lot of work to show the safety of this, and eventually we got approval from the UK government regulatory agency, the MHRA, which works on behalf of the EMEA in the UK. They approved our trial just under 1 year ago, and we've now started that trial. It's a safety trial in 10 patients, but we'll be looking for efficacy, whether it works or not, at the same time. So that is underway. We hope to be able to give an initial report in about a year's time, and the formal follow-up is at 2 years. And if that's looking promising, then we'll go on to a larger trial within 4 to 5 years from now. So I've talked about sheets, and I've talked about healing tears or gaps between tissues. I also talked about tubes, and how, because tubes in the body are relatively thin, they're really just sheets that have curved round, so the principles are the same as for sheets, really. And this is an answer paper that you mentioned that we published in November 2008, and the reason it caused so much fuss in the media was it was the first clinical application of tissue-engineered airway replacement, and it was kind of seen as the first example of a life being saved from an engineered organ. Whether that's really true or not, you could debate. There's all sorts of issues around that, but that's how the press saw it at the time. So the connection with the earlier work I've described, you may realise, is that the airway, the trachea, has cartilage in it, C-shaped rings of cartilage, which are really important for the function of this airway. Without it, the airway would collapse under the pressure of the air flowing in and out. Or if it was too rigid, then you wouldn't be able to move your chest and so on. So cartilage is important to the airway. The patient, and I can give you her name because she's been named in all the newspapers and she's given us permission to talk about it, she's called Claudia Castillo. She grew up in Colombia, where she developed tuberculosis that was not properly diagnosed. It was mistreated and it spread through her airway. She had an upper airway lesion that was treated effectively using conventional medicine, but she had a lesion in her left bronchus, that's the first branch of the trachea, that was badly damaged. It was treated with a stent, which is a plastic or metal tube that's inserted, but the stent did not work and had to be removed 6 months later. That's often the problem. She was a 29-year-old single mother. She was getting worse and worse. The surgeon, Paolo Macchiarini, now she moved to Barcelona, he was as certain as he could be that she was going to die. There was a possibility to remove her left lung and left bronchus, but he felt that it was very unlikely that she would survive that operation. You can see, I hope, the lesion here. So this is the trachea, the airway. This is the right branch, which you see is nice and open. And this is the left branch, and I hope you can see how constricted it is there. And on the video on the next slide, you'll see we're looking down her airway and we'll see that lesion. So here's the open right bronchus, the right branch, and this is the closed. You see how closed up that is, the left bronchus. There was more or less no air getting in through there at all, and that was really making her very ill indeed. That lesion went on for about 6 centimeters, and the animal work that Paolo Macchiarini had been doing in pigs to try out this technology, you suggested that up to 4 centimeters could be treated with one particular technique, which I'll come to, but above 4 centimeters would fail. So the principle was to use decellularized tracheal tissue, so to take airway tissue from a donor and remove the donor cells. And the reason to do that was to prevent immune rejection, so that the patient would not need immune suppressive therapy for the rest of their lives. And what Paolo showed in his pig study is that decellularized airway, which is essentially just a material with no living cells, worked in small lesions up to 4 centimeters, but not bigger. He and we hypothesized that if we wanted to do larger, longer pieces of trachea, we would need to put cells back on, and to prevent immune rejection, that should be the patient's own cells. So the idea here was to take donated tracheal tissue, remove the donor cells, and replace with cartilage cells that we would grow up in Bristol on the outside, and with epithelial cells, which is the cells on the inner lining of the airway, grown up from a biopsy from the patient's airway on the inside. And there were different techniques across Europe that might be used to do this. One technique for removing cells from tracheal tissue, our technique for making cartilage cells from stem cells, which I haven't gone into today, but we've been working on those. My colleague Martin Birchall's technique for growing up airway epithelial cells, something called a bioreactor, which I'll show you, a machine for culturing all of these together, the cells seeded onto this scaffold, this tube, for 48 hours to get the cells to grow in. And then the surgical implantation methodology Paolo had been developing. When Martin rang me in March 2008 and said, Anthony, we want to save this patient's life and you're going to help us, I thought he was being stupid, and I told him, don't be daft, we have no idea whether this science can work at all. But eventually I was persuaded that this patient was very, very ill, was probably going to die, and there was just a chance we could make this work. And I realized that I had been saying for a long time regenerative medicine is going to save lives, and actually here was a patient whose life needed saving, and just possibly it could work. It was a risk for us, not a risk for the patient because she was probably going to die anyway, a risk for us reputationally. We had to try this, and if she had died, even though she was going to die anyway, I think reputationally it would have been quite damaging, but I and the other members of the team felt we should give this a go. The legal authorities and the regulators really didn't know what to do with us, so they let us get on with it and would have then told us it was all our fault if it had gone wrong, but luckily it didn't. So we went ahead. So the general approach again was to take a iliac crest biopsy from the hip, from Claudia's hip, grow up her stem cells, seed them onto the donated tracheal tissue with the epithelial cells in the middle, and create this, what I call approximation to a trachea. So this is a diagram of a normal trachea with C-shaped rings. What we were doing was not making C-shaped rings, but creating cartilage all the way down the full length of the 6 centimeters of this piece of tissue we were creating, and then putting these cells called epithelial cells, which are specialized cells, down the middle to create a kind of barrier that would also drive vascularization on the inside. So cartilage on the outside, epithelium on the inside. That was the idea. And again, it's a really important point. In tissue engineering and regenerative medicine, we don't necessarily need to make a perfect tissue that looks and behaves exactly like the natural tissue. We think that approximations to a tissue in some cases will work okay. So, the trachea was obtained, and it was treated gently with these detergents to remove the donor cells to avoid immune rejection. Here's the, at the beginning with all the cells still in there, the little dots in there. And here at the end, there's none of those little dots because all the cells have been removed. And for those who know about MHC antigens and the immune antigens that drive immune rejection, they're stained in brown here at the beginning and they're absent at the end of this process. So we were relatively sure that we produced a tube 6 centimeters long that was no longer had donated cells on there, though we couldn't be absolutely certain of that, and that it had mechanical properties that were unaltered compared with the natural tissue. That's shown using pig hand. In Bristol, we grew up the mesenchymal stem cells. MSC means mesenchymal stem cell. It's just a shorthand for this particular kind of bone marrow stem cell, using a particular regime that we adapted from some of the work we were doing, and it's not rocket science. This kind of work was going on all around the world, although we had fiddled around with some of the growth factors in a way which we felt was beneficial. In Bristol also we grew up these epithelial cells. Then we had to transport these cells from Bristol back to Barcelona in order to get them onto the decellularized scaffold and implanted into the patient. And that created a bit of a problem, an unexpected one. So there's only one airline that flies between Bristol and Barcelona, and that's EasyJet. Martin Birchall, this surgeon, ran EasyJet head office in the UK every day for a week before the day we knew we were going to have to transport these cells. And he said, we've got these cells, we need to get them to Barcelona, we're saving the patient's life, we need to make sure you're okay with that. And every day head office said, yes, it's absolutely fine. On the day of the operation, a young trainee German thoracic surgeon working in Barcelona with Paolo Macchiarini flew over to Bristol, collected the cells, went to check in. You can't take those on board, sir. Why not? Because it was more than 100 mils of liquid. There you go. Martin, the ENT surgeon, he's about that high, but he got really angry. He was shouting and arguing with the armed police were called along. He was about to be arrested. So the German thoracic surgeon said, right, let's calm down, let's just talk about it. And they ruled out another airline and get the same response. They ruled out driving, it would take far too long and the cells would die. And then the surgeon said, I know a thoracic surgeon in Germany and he flies planes. Why don't we ask him? So EasyJet having failed, they called this guy who had trained with Paolo Macchiarini, and he said, yes, I have a small plane and a big plane, which one do you want? He flew over in his small plane to Bristol, picked up the cells and took them to Barcelona. Otherwise, we wouldn't be here talking about this. Total cost €10,000, which EasyJet refused to pay, but they gave us the price of the ticket back, which was about 50 euros. Okay, they get to Barcelona, and here is the actual decellularized scaffold with the epithelial cells already on the inside, and it's put onto this plastic roller, and this is it's just a box. It's called a bioreactor, but it's a posh name for a box that does things. And the chondrocytes, with the cartilage cells made from the stem cells, were carefully pipetted along the length of this, but it's really not sophisticated science. This was real cottage industry stuff. And the bioreactor has an air and a medium component to try and simulate the airway, and it was grown in there for 48 hours, and then on the 12th of June 2008, the patient was operated, and the diseased bronchus was taken off and replaced with the tissue-engineered trachea. And here it is just before implantation, and here it is immediately after implantation. You can see that's the natural tissue that's red. This is engineered, so it's white. There's no vasculature at that point because it's just been implanted. So the next video is looking down through this airway on the day of implantation. And it will start, here it is, and you can see again it's white, there's no vasculature, but the important point is that air is flowing very nicely in and out of that from the day it was implanted. It never leaked air, and her airway function has remained normal ever since. And 1 month later, you can see here not only is air still flowing through, but you see how red that is, and we think the epithelial cells that were seeded onto the inside have driven an angiogenic response. That's the hypothesis at any rate. And we took a biopsy at that point, and there was clear evidence of mucosal reformation. That is, the barrier that prevents infection and protects that inner surface was already growing back, and I think that's a very important part of why this survived. So here you can see now the left bronchus is nice and open, equal in diameter to the right bronchus. So to summarise, for this patient, the tissue-engineered airway was patent. That means it didn't leak air from the moment of implantation, and her airway function has been excellent ever since. At 1 month, there was evidence of mucosal formation and revascularization. She's never shown any signs or symptoms of immune rejection. She's never been on immune suppressive therapy, and she's no longer in healthcare. She's back to work as a dental hygienist. Would she have died without this operation if she had just had her left lung removed, left bronchus? Of course, we'll never know. The surgeon's view was she was almost certainly going to die. It's not a laboratory experiment, we can't ever know, but our collective view was she probably would have died, and now she's alive. So if you accept that at face value at the moment, then I hope you'll see in this kind of case that regenerative medicine can remove the disease from the patient and get them back to where they were before they were ill, and that's quite a powerful outcome. Okay, so that's one tube. This, very briefly, I'll talk about a study from Tony Atala's lab in Wake Forest in the USA. So nothing to do with my laboratory, but it's another example of a tube. This was published in 2011, and this was repair of the urethra, which is the tube connecting the bladder to the outside world in children with traumatic injury to that part of the anatomy, and so they were incontinent, which meant they were leaking urine, which for kids with the whole of their lives ahead of them is a really unpleasant and nasty problem to have to deal with day to day. And this team used a biomaterial to create a tube shape which they coated with urothelial cells, which are the specialized cells that were needed, a little bit like the chondrocytes, the cartilage cells that we implanted on the trachea. Urothelial cells on the inside and smooth muscle cells on the outside. And I'm not going to go through this because I'm in no way an expert on this part of the anatomy, other than to say the Lancet paper reported very good results several years after implantation. So it's another example of a tube where it seemed relatively easy to get cells in there and implant. This wasn't decellularized donor tissue in this case, it was an artificial material combined with cells. Another kind of tube in the body are blood vessels, and there are many groups around the world working on how to engineer blood vessels, and again, because they're tubes, we think they're relatively easy to produce, although these are tubes that usually need to move, they need to contract and relax, and so they need smooth muscle function, and sometimes they've got valves in them, so they're a bit more complicated. I'm just using this to introduce to you a project we've just started, I say just 3 years ago, that we're well into. So this is a video from my colleague Massimo Caputo in Bristol, who's a pediatric cardiac surgeon. So he treats babies with hole in the heart and other kinds of heart, congenital, genetically inherited heart disease. And here he's implanting decellularized donated blood vessel. It may be human, it may be pig, they use both. And implanting that into this baby. And that's a very good, very successful operation that saves the lives of these babies within a few days of their being born. So here it is being put in place. Great operation, life-saving, but there's a problem, which is, you see how big this is? That's bigger than it should be for this size baby, but the babies grow so quickly that that relatively large size actually is too small for the baby as it grows, and so these kids have to be reoperated often 2, 3, sometimes even 4 times through their lifetime, and that's very traumatic for them. Can be serious, because any surgery of this sort is serious, life-threatening, and really gives them poor quality of life. And what we want to be able to do is, well, our hypothesis is that if instead of implanting decellularized vascular tissue, we use cells seeded onto the outside, smooth muscle cells onto the outside of those blood vessels and endothelial cells on the inside, that those blood vessels may then remodel and grow as the baby and the child grows. That's the hypothesis. And the cells we want to access for that are the umbilical cord stem cells, which we think we can turn into vascular, smooth muscle cells. So it's early days. I can in no way say we're going to get this into the clinic. We're still doing a lot of the very basic science to try and make this work. But it's another tube. Okay. What about solid organs? Can we get anywhere near to creating large organs? The heart is a relatively large organ with a great big massive muscle. And when we first did the trachea, journalists would say, well, you know, how far can you take this? I said, well, you know, don't think about the heart or the liver or the kidney. It's a complete waste of time. And then I went back and did my homework on the literature, which I should have known because it was published just before we did our work, but I was not so familiar with this paper at that time. I became very familiar with it from Dorothy Taylor's lab in Detroit. And here they showed that they could decellularize rat hearts and they could repopulate them with stem cells and get some beating activity to those hearts. Now, they're rat hearts, so they're quite small. It's not the same as the human heart. And it wasn't full activity, so we don't know if this can ever be used as an approach to create a really functioning heart. But I think the principle is there that by decellularizing you can retain the shape of a complex organ, and that gives us a chance of moving into that kind of tissue. So if we look at what's next, I think we should be thinking about what we need to do to move on from sheets and tubes and healing lesions and think about complex organs like kidneys and liver and heart. If regenerative medicine is about anything, it's got to be about trying to solve some of these problems. This is from that Dorothy Taylor paper showing the decellularisation. Here's the ratheart before, and then as it's gradually decellularised, and the detergents to decellularize are pumped through the heart vasculature. You can see it becomes completely translucent, but the vascular tree remains intact, which is really important, because then it can be recellularized through that vascular tree. So the vascular tree is the supply of blood vessels throughout the heart. So here you can see it being decellularized, but with the vascular tree intact. So decellularization is possible, but there's a real problem in principle to large organs, and that is, how do you get oxygen into them? I said to you before, I thought that thin tubes and thin sheets were relatively easy because you can get oxygen into those cells while the organ is growing. And this is an experiment we did in my laboratory just to try and test if oxygen supply is a problem. Even for cartilage, and cartilage cells don't usually need oxygen, or not very much, to grow. What we're doing here is comparing two scaffolds, so pieces of material, this is polyglycolic acid, loaded with an appropriate number of cells for the size of the scaffold, but up here, these are thin scaffolds, just 2 millimeters in thickness, and here in this row they're thick, they're 6 millimeters, so 3 times the thickness. And what you can see here, let's just focus on this. This is staining the cartilage that's been made after a month. And you can see on the thin scaffolds, that's fairly evenly distributed across the whole of the scaffold. But in the thick scaffolds, you get this outer ring, and there are not many cells and not much cartilage in the middle. And where we believe that that's because of lack of oxygen, and the evidence for that is that those cells that are there, which you can see in this ring here, this is staining for something called HIF1 alpha. Don't worry about what that is, except to say it marks out those cells that are deprived of oxygen. So you think these cells have burnt up all the oxygen around the outside, and there's just not enough getting into the center of these thick scaffolds to allow cartilage to form. It's just one experiment. There's other evidence that oxygen is important, but it's not the only important issue in growing large organs. There's oxygen, there's shape. Decellularization may help us to make complex shapes. There's immunity. How do we create a complex organ with many different cell types and avoid immune rejection? And there's those cell types. Kidney has many different cell types in, for example, each with a different function. How on earth are we going to do that? I don't have answers for you. I cannot say to you there's a magic answer, which is this is how we're going to solve this problem, how to make complex organs. My message to you, and to those of you who are interested, and to those of you scientists who are working in this field, is these are soluble problems. If you look at it as an engineering problem, we can break it down into these different component parts and find different technologies to solve these problems and put that back together in a way which will allow us to do things we can only really dream about at the moment. And some of those technologies are already there or thereabouts. So, for example, we are developing in my labs a technique called cell painting, which we think we're going to be able to use to deliver oxygen very close to cells and keep it there at quite a high level during the tissue engineering process. And we believe that that's going to help us solve these problems. There's a technique that's being used in many labs now called 3D printing, which allows the creation of different layers of scaffold and cells so that you can grow up a whole solid organ by creating lots of different sheets of organ and bringing them, of tissue and bringing them together. 3D printing is, if you like, an alternative to the decellularization approach I've been telling you about. And then the problem of immunity and of different cell types could both potentially be solved by these things called iPS cells that some of you will know about. iPS is induced pluripotent stem cells, a technique discovered by Yamanaka and his colleagues in Kyoto. He won the Nobel Prize. This year was a co-winner of the Nobel Prize for this work, where he could take any of our cells, let's say our skin cells, and reprogram them by inserting 4 genes to turn them into something that's like the most primitive stem cell you can find, the embryonic stem cell. And although it's early days and there are particular safety and technological issues in understanding how to use these cells, I do think they offer a real way forward, and without any of the ethical dilemmas that are faced by the use of embryonic stem cells that many people are worried about. So although the scientific community is relatively split on whether iPS cells will be the answer to all these problems, it's certainly one possible answer in many tissue engineering situations. So I think these are what I call building blocks for thinking big, for growing these large, complex organs, and by picking away at each of these problems, developing these technologies, developing other technologies, that we really will be able to make advances and move on from these flat sheets and tubes and into more solid organs. I'm going to leave you with Claudia. This is the patient who we treated in Barcelona, and I had the pleasure of meeting her 3 months after her operation. And she doesn't look very sick now, if you see. And through a translator, because she doesn't speak English, she's talking about how her kids are playing in the playground there, how now she can play with them when she couldn't before. She could live a normal life. I don't think she was working at that time, but she is now. And she was thanking the team and so on. I'm a scientist. I run a lab, I do my science, and for me to come face to face with a patient in whose chest is a 6 centimetre segment of airway, engineered using cells grown up in Bristol, using techniques we've developed over the years, it was a very moving moment and brought home to me how, even though our science isn't perfect, nowhere near perfect, nevertheless, we as a scientific community collectively can have impact on people's lives, and I hope the scientists in the audience will share with me the excitement of that knowledge. Not a reason to cut corners and rush, but it is a reason to really drive home our science and make it work. And that's the last. Thank you very much.